decided to do the usual study update for Revive in next week's presentation, so that will not be done today. So we are going to jump in and we are going to welcome the honored Professor Nick Feasey as our keynote speaker today. Prof. Feasey is a leading clinical microbiologist and infectious diseases specialist, holding appointments at the University of St. Andrews and the Liverpool School of Tropical Medicine, as well as serving as an NIHR Professor of Global Health. He currently practices as a consultant microbiologist and has previously held senior clinical posts at the Royal Liverpool University Hospital and Queen Elizabeth Central Hospital in Malawi. His research centers on antimicrobial resistant bacterial infections with a strong One Health approach to understanding the transmission of enteric pathogens that drive invasive disease. He's deeply committed to improving healthcare-associated infection control, particularly through infection prevention and control programs in southern Malawi. Prof. Feasey integrates implementation research, health economics, bacterial genomics, spatial statistics, and transmission modeling in close collaboration with global partners, including the Wellcome Sanger Institute and the University of Lancaster. We are delighted to have him today, and we look forward to his insights. Prof. Feasey, over to you.
Thank you very much. Thank you for the welcome. Hello to old friends and new on the call. Let me try to share my screen. Right, that should be in standard view. Can you now see my talk?
Yes, we can. Thank you.
Okay.
It's not in, sorry, Prof. Feasey, it's not in presenter mode yet.
Let me see if I can. I think it's because I'm using a two-screen setup. Is that better?
It is still the same, not yet.
Is that better?
Yes, that's perfect.
Cool, thank you. Good afternoon, everyone. I've been invited to speak to you today about clinical implications of antibiotic resistance in Malawi, where I lived and worked for many years. And thank you to Sean Wasserman and Graham Minkis for the invitation. And thank you to Elsa for all of the organization and for the thorough introduction. The data that I'm going to present today largely comes from the Malawi Liverpool Wellcome Research Programme on the site of Queen Elizabeth Central Hospital in Blantyre, Malawi. MLW, as many of you will know, is one of the 5 Wellcome major international programs. In this picture, you can see Blantyre, the second city of Malawi in the southern region of Malawi. And MLW is on the site of Queen Elizabeth Central Hospital, which is still just about the largest hospital in Malawi, but also its location highlights the long-standing commitment to supporting translational healthcare research in Malawi. MLW is an affiliate of the Kamuzu University of Health Sciences in Malawi, which is one of our key partners. So we work very closely both with KUHAS and with the Ministry of Health. It was originally established as a malaria pathogenesis research institution, and it was just Cecilia.
Sorry, I'm gonna mute Cecilia. I'm just gonna mute you.
It was originally established as a malaria pathogenesis research institute, and Malcolm Molyneux, the founder, was quick to identify that in order to study malaria effectively, it's very important to have blood culture capacity. So we are fortunate to have a very high quality research institute on the grounds of what is in many respects a very typical sub-Saharan African hospital. And in the picture on the right, top right hand corner, you can see both that the wards are often crowded, and also the people sitting on the ends of beds are guardians. So an awful lot of healthcare is still delivered by close family members or close friends. Malawi is a low-income African country, and the GDP is still less than $1,000 per head of population per year. The life expectancy is about 64, and that represents an incredible success. In 2005, life expectancy was 44. What that means is there has been a rapid transition to the importance of non-communicable diseases. Conditions like stroke are starting to become more important. This is leading to longer hospital stays and, of course, longer interactions with the healthcare setting. You can see Malawi placed in the context of some of the other welcome major international programs. And Norway is an example of one of the highest income countries in the world. So what does poverty mean in the Malawian setting? And there's examples of a photo from a very rural setting and a high density urban setting. It means there is day-to-day precarity in the lives of people. It is still the case that about 75% of Malawians are subsistence farmers who struggle with severe soil erosion. There's approximately a 9% HIV prevalence, and severe infections are common. And straight away, I want to say that antibiotic usage in Malawi is often a rational response to a severe burden of severe bacterial infection. And I don't like sometimes slightly lazy stereotypes about inappropriate antibiotic usage. The financial situation in Malawi has deteriorated in recent years, initially following the COVID pandemic and more recently following the war in Ukraine, because so much fertilizer globally comes out of either the Ukraine or Russia. So the cost of a bag of fertilizer, which is essential for subsistence farming, has gone up by at least a factor of 5 in recent years. Healthcare provision is free at the point of delivery. However, the investment is about $39 per person per year, and about 3/4 of that is aid. And of course, aid budgets have recently been critically reduced this year in particular. It also means that there is an unequaled opportunity for impact. You can see in the graph on the right, the places where currently life expectancy is lower and GDP is lower, you can see are on a steep inflection point where investment in healthcare has the greatest opportunity for impact. You can also see in the left-hand side how life expectancy in Malawi recovered from the peak of the HIV and has gone well beyond that and resuming its previous upward trajectory. So I mentioned that Malcolm Molyneux established blood culture capacity. This is one of my PhD students many years ago now, a lecturer, Dr. Patrick Musicha, who looked at our first 20 years of bacteremia surveillance. So at MLW, blood cultures were done on, or have been done on unselected populations of adult medical patients and pediatric medical patients. And the image on the left represents the summary of our first 200,000 blood cultures. Now you can see the red bars on the left-hand side, the general gentle decline of significant pathogens subsequent to 2005, when there were many important public health initiatives around HIV control, malnutrition control, and malaria control, which all contributed to improvements of health in the population. On the right-hand side, though, you can see a slightly grimmer story, the rise in the number of untreatable isolates, given that we have often struggled to access we are dependent on third-generation cephalosporins, and we often struggle to access carbapenems. So pathogens are going down, but antimicrobial resistance is certainly going up, particularly in very vulnerable populations such as neonates. The bug-drug combination, of course, only tells half of the story. So over the last few years, we have little by little been trying to dig into what this actually means. So here you can see a subsequent PhD student of mine, Joe Lewis, who's now a senior lecturer at the Liverpool School of Tropical Medicine. And in his Wellcome-funded clinical PhD, he endeavored to assess what is the burden of severe infection in adult patients, both looking at microbial causes, but also looking at the impact of therapy, typically with third-generation cephalosporins, on acquisition of antimicrobial resistance. Ultimately with an aim of developing an antimicrobial strategy for severe infection. And that's something that I continue to work on and currently discussing with Sean and Graham. Now, he recruited 225 patients meeting the sepsis 2 definitions. This study started just before the launch of sepsis 3. 87% of whom received ceftriaxone. And he found that disseminated TB dominates as a cause of sepsis. And as we all know, third generation cephalosporins do not cut the mustard in the treatment of tuberculosis. He also found that empiric TB therapy is associated with survival in a propensity score matched analysis. But broadly speaking, in the plot on the left-hand side, you can see actually not very much that would respond to a third generation cephalosporin. I think about 15% of patients had a positive blood culture. And about 2 individuals had an ESBL producing one. So antimicrobial resistance at the point of presentation in adult medical patients was not a major problem. However, third generation cephalosporin non-susceptible etiologies was. He then decided to look at the impact of current sepsis management in Malawi and how that contributes to the acquisition and carriage of ESBL producing coliforms out of the hospital. Now, in the red bar on the top here, you can see adults admitted with a need for third generation cephalosporins. And in the blue bar, you can see adults admitted with no antibiotics given. And the green bar is the healthy population. And it is striking that about 30% of participants were already, the healthy participants were already carrying ESBLs in the community. Amongst people who were hospitalized and didn't receive ceftriaxone, you can see a slight blip in ESBL carriage, which then rapidly declined. And then this really impressive bulge in response to hospital exposure and third generation cephalosporins. So here the modeling and the simulation suggest synergistic effects of hospitalization and antimicrobial exposure with a prolonged post-antibiotic effect. So people coming in, getting antibiotics that are perhaps not indicated, are then fecally excreting ESBLs for prolonged periods of time. The second question we tried to address, and this was a Wellcome Clinical PhD led by Rebecca Lester, who's now a senior clinical lecturer at UCL, was what is the burden of ESBL producing bloodstream infection in Malawi? And she started off with a systematic review of this problem in which she identified that in Africa, the prevalence of ESBL producing bloodstream infections is high, but in 2020, when this was published, the clinical outcomes were largely unknown. So she recruited a longitudinal cohort at Queen Elizabeth Central Hospital to answer this, trying to assess mortality and quality of life. Now, she found that the outcomes from culture-confirmed gram-negative bloodstream infection in Malawi were poor, even in the presence of a cephalosporin susceptible bug. So you can see a 34% case fatality rate, but this did significantly rise in those individuals with ESBL producing bloodstream infection, although perhaps not quite as much as one might have predicted. But certainly the presence of resistant bloodstream infection was associated with increased in-hospital mortality. She also evaluated health economic outcomes. So she looked at healthcare resource use in order to infer direct medical costs, patient level costs to look at direct non-medical and indirect costs to produce a societal cost. And it was estimated that it costs an extra $81 to treat a patient with third generation cephalosporin resistant bloodstream infection, and that patients spend significantly more money. So the additional cost to society using this economic modeling was an additional $230. So we decided next to try to investigate where these drug-resistant infections were coming from. And here is Derek Cocker, yet another Wellcome Clinical PhD student, now a lecturer at the University of Liverpool, who led the Drivers of Resistance in Uganda and Malawi project in Blantyre. Now, the purpose of DRUM was to evaluate community carriage of these ESBL bugs and to try to identify where they were coming from and going to. And in a piece of community engagement undertaken with and led by partners in Uganda, The cartoon here was produced, and it was decided to place households at the center of this study to focus on ESBLs as a key resistant determinant, given the importance of third-generation cephalosporins in both Malawi and Uganda, and to focus on E. coli as a ubiquitous pathogen in the community, and Klebsiella pneumoniae as a bug that is more closely associated with healthcare-associated infection. This was just one work package of several in Drum, which was MRC and MRC and NIHR program grant. And this particular program looked at investigating how human health, antibiotic usage, and water sanitation and hygiene acted as drivers of antimicrobial usage. And so Derek employed a community-based household study design, sampling in urban, peri-urban, and rural areas of Malawi. And there was a parallel study done in Uganda. And the reporting from that is somewhat delayed, as all of this was going on ultimately at the peak of the COVID pandemic. And the COVID-related lockdowns in Uganda were much more strict than they were in Malawi. So it was easier for the Malawian sites to proceed. We sampled all households really quite intensively from a microbiological perspective, but undertook in-depth water sanitation hygiene observations in a selection of them over a 6-month period of follow-up. We took external environmental samples. We sampled river water. We took a bunch of different samples from the household environment, and from human household members, we sampled stool. The water sanitation and hygiene evaluation methods looked at collecting demographics. They made multiple observations, undertook a semi-quantitative survey called the RANAS survey, leveraged in-depth interviews with key informants, and undertook transect walks mapping high-risk areas, and then went through results validation sessions in partnership with the communities in which we were working. Household selection was via a process called an inhibitory close pair design, and this household selection was led by colleagues at the University of Lancaster. But it was to account for any potential spatial signal. In total, we recruited 600, we screened 611 households, I apologize. We recruited 300 and undertook 300 baseline visits. And 195 households were allocated to long-term follow-up. So what did we find? And the photos taken in this presentation were by someone called Tokozani Chikondi, who is a photojournalist with consent for use by the participants, but I thank Toko for her excellent work. So the average house size was 4 people. Only 2.3% of people live above the poverty line. Most households owned animals, especially poultry, particularly in urban settings, and cohabitation with domestic animals was particularly seen in urban settings. And this was explained to us by participants as a reason of protecting the animals from theft at night time. So, and again, you can see in this photo that inadequate sanitation infrastructure was present in the photo and common overall. So this one photo gives many examples of systems that were present in different ways across urban, peri-urban, and rural settings in Malawi that do not promote good and safe environmental hygiene. Interestingly, and this has been replicated in other settings as well, that community antibiotic usage was limited in spectrum and fairly limited in quantity as well. So about 15% of participants had received an antibiotic in the previous 6 months, but this was commonly limited to 3 oral antibiotics: cotrimoxazole, which was widely available from the HIV program, amoxicillin, and metronidazole. And again, these 3 antibiotics have commonly been seen in other settings as well, thinking about Claire Chandler's work in Uganda. Children under 5 and participants living in rural settings were actually more likely to have received an antibiotic for reasons that we don't fully understand. In contrast, and clinicians do love to blame vets, but in contrast, there wasn't widespread animal usage of antibiotics at all. So at least 70% of households owned at least one animal, and disease and illness episodes were commonly reported, particularly in poultry, but less than 1% of animals received an antibiotic from within the households that we studied. And again, this was explained as being because actually human antibiotics were cheaper to the people who needed them than animal antibiotics. In terms of water sanitation and hygiene factors that potentially drive antimicrobial usage, hand hygiene was identified, water usage, and sanitation and waste management and people's environmental interactions were all identified as potential risk factors for acquisition of enteric pathogens, enteric ESBL pathogens. So what did we find in terms of the ESBL? So there was a high level of ESBL producing coliforms found in these households. So about 40% of humans, so even more than Joe Lewis found, were ESBL carriers. This was mirrored in the co-located animals, and really high rates of ESBLs making their way into the drains, leaving people's homes, and then into the river systems of Blantyre. Blantyre in particular has many rivers running through it. It's on a plateau between three mountains. There was some site-based difference identified statistically. However, in practice, the numbers weren't that different. It's just the numbers were very large. And there was some species difference in animal colonization as well. So starting to look at this longitudinally, we found that human ESBL colonization fluctuated. And in this facet plot from the Ndurandi region of Blantyre, you can see all of the households summarized with the individuals and the time. So ESBL colonization fluctuated between households, between individuals, and also between regions of the study. But you can see that the dark blue being ESBL positivity, there wasn't a single household that didn't have a person colonized at some point. In terms of co-located animals linked to households we were studying, we found that presence varied by animal species, with pigs and poultry having the highest colonization rates and poultry being present in particularly high numbers. So the One Health aspect clearly is important here. There was widespread environmental ESBL contamination. So these ESBLs were found widely throughout household surfaces. They depended a little bit on the water source that household water was obtained from, and a high prevalence, as previously mentioned, in the drain water in all of the areas, with a high prevalence in river water. We found that human ESBL colonization was linked to seasonal effects and exposures with a heavily contaminated environment. So in the case of E. coli, the key risk factors were urbanization, animal interactions, and humans with advanced age. There were different patterns for Klebsiella, where increased household density, evidence of exposure to drains, and human fecal contamination seemed to be more important. But what was common was that prevalence increased in the wet season. So we now are at the point of having a huge and incredibly complicated data set, and we wanted to understand how to make sense of things better. And so we decided to characterize the bacteria using whole genome sequencing and using statistical modeling. This is Lorenzo Rimella, who was a postdoc at the University of Lancaster. And he highlighted, using his modeling approaches, the importance of location and climate. So in the three maps of Ndurandi, Chileka, and Chikwawa, you can see that in all three settings, geographical hotspots of antimicrobial resistance existed. His modeling really confirmed the previous signal that there was a stronger E. coli association with the environment, whereas the model supported K. pneumoniae transmission via person-to-person contact. And again, the modeling highlighted the importance of seasonal changes in transmission dynamics throughout the year. So the next step was to undertake whole genome sequencing. And again, this is Patrick Masicha, this time in his role as the postdoctoral fellow on drum when he was based at the Wellcome Sanger Institute. He's now the AMR research lead at the Malawi Liverpool Wellcome Programme. And in this radial phylogeny, you can see the outer ring represents country, with green being Malawian isolates and the kind of lime green being Ugandan isolates. And the inner ring is the source, whether the bacteria from humans, animals, and the environment. So there is an awful lot of diversity represented in this tree. And if I draw your attention to the 12 o'clock position in the radial phylogram where ST131 are highlighted. So for people who are not E. coli enthusiasts, ST131 is a lineage of E. coli that is particularly associated with antimicrobial resistance and ESBL production that was first identified about 25 years ago and is now a globally dominant lineage of E. coli. But here there is a surprising amount of structure to this tree, which is particularly driven by the Ugandan isolates. Given the global importance of ST131 and the importance of ST131 in our studies, Patrick zeroed in his work on building transmission networks of this bug. Now, we are all continually told that antimicrobial resistance is a One Health problem. Quite often people don't go the next step and then drill into the relative importance of One Health for different bacteria. And quite often when this work has been undertaken, it hasn't necessarily supported the dogma. But here, by reconstructing the transmission networks based on ST1 genomes, Patrick has elegantly described that there is flux of ST131 E. coli between humans, animals, and the environment. And this paper should come out in the next couple of days in the Lancet Microbe, really highlighting how in the Ugandan and Malawian settings, ESBL producing ST131 E. coli really are a One Health problem. And I think these are important data that Patrick has generated. So, to summarize, there is a high prevalence of ESBLs found in human and animal stool and in the broader environment. The key risks for human colonization with ESBLs are the wet season, urbanization, and animal food interactions. ESBL E. coli isolates cluster independent of ecological source, indicating they are very much a One Health problem. And rivers are heavily contaminated with antimicrobial resistant bacteria, antibiotics, and resistance-driving chemicals. So a One Health approach is clearly needed to address the challenge of antimicrobial resistance in Malawi and similar low-income settings. There are very many people to thank, and I did highlight several of them as I went along, but key PIs, Chris Jewell at the University of Lancaster, Tracy Morse at the University of Strathclyde, Kondwani Chidziwisano at the Malawi University of Business and Science, and Nick Thompson at the Wellcome Sanger Institute. And there are many other people highlighted in the attached slide. Also very important to thank the key funders of this program grant, the MRC part of UKRI and the NIHR, but the core funding from MLW comes from Wellcome, and my laboratory in Malawi was established thanks to funding from the Bill and Melinda Gates Foundation. So I will stop there and stop sharing, and very happy to take questions. Thank you very much for listening. I can see Graham. Hi, Graham.
Graham, you want to go first?
Yeah, no, thanks. Thanks, Nick. Sorry I joined a little bit late, but really exceptional body of work, and thanks for sharing that. I just got two questions. The one is, compared to our experience in South Africa with ESBLs, and I'm not aware of sort of community-based studies like you've done in Malawi, but just as a clinician working in a hospital setting, finding ESBLs is so strongly linked to recent hospitalization rather than community transmission. I'm just wondering, do you think this is unique to Malawi, and or is it something that has been studied elsewhere in Africa, and is it very much related to the kind of sanitation systems in those countries that might be driving this? Because it is just such a strong difference to what we see in South Africa, for instance. And maybe I'll follow on with the second question.
Thanks, Graham. So I think there is an absolutely profound difference between sanitation infrastructure in Cape Town and much of South Africa compared to an extremely poor country such as Malawi. So yes, I think those barriers which create evolutionary bottlenecks between the different One Health compartments just don't exist in Malawi in the same way they do in other sites. And I think there are, I mean, I didn't present the Uganda data because there are Ugandans working on it, but the signal is similar from the Ugandan study sites, and I've seen similar data from Kenyan groups as well. So I think this is the impact of lack of sanitation. And one of the reasons this is important is because it's an example of AMR being a very tractable marker of failure in infrastructure. These were carried isolates that we were studying in drum rather than disease isolates. And really what we are seeing here is that because the ESBL determinant makes it really easy to culture these samples, it therefore makes it easy to highlight this particular set of problems.
Yeah, and then Nick, the other question was that data that you showed of ESBL infections in a hospital setting, and the mortality overall for bloodstream infections was about 35%, but yet it was worse with ESBLs, but perhaps not as bad as one would expect. And how were those? Firstly, your explanation for that, and secondly, and related, how were those patients treated? What antibiotics did they have access to?
Thanks. Again, really important question. I think one of the things that the antimicrobial resistance community are obsessed with bugs and genes, and an intensive care physician might say to you that antimicrobial resistance has never killed anyone, but a final common pathway, which is sepsis, is killing them. And I think in the Malawian context, there are very many excellent doctors who are let down by a lack of funding to deliver high quality care. And how that plays out in sepsis is a lack of high dependency care beds, lack of intensive care beds, and actually, as you and I have discussed before, a lack of understanding of the most appropriate physiological response in terms of fluid resuscitation and use of inotropes. So in other settings, yes, early deployment of antimicrobial chemotherapy is important, but so is physiological stabilization. And we're only really able to address the antibiotic side in Malawi. So I think the case fatality rate in people with drug susceptible sepsis is inflated by the lack of adequate physiological support, and that's why I think that there is that closeness.
But to kind of ask the question another way around, the survival among those with ESBLs, is there access to carbapenems, etc., for those people where it's diagnosed?
Infrequent is the answer. Quite a lot of the patients in that group will be driven by the neonatal sepsis cohort, and there is slightly greater access to carbapenems and to amikacin in the neonatal group. I think one would probably expect a survival of about 70%, a case fatality rate of about 70% in people who didn't receive antimicrobial chemotherapy. So there's a very live question in the AMR community at the moment as to whether or not there is some level of activity of the agents that we use in people with AMR infection, because it's a problem that nobody can completely explain.
Yeah, so that, I mean, these are people with bloodstream infection with a resistant organism treated with antibiotic to which they're resistant, but 1/3 of them actually survive and do well.
Yes.
Yeah. Okay. No, thanks. That's really useful. Thanks.
There was a question from Helen, and then it can be followed by a question from Thomas.
Thanks. A very fascinating talk. I find it particularly interesting because we're busy with looking at a bit of a similar study in a rural area in Eastern Cape. And there, in fact, there's no running water. So we might find more similar findings to what you find in Malawi than would be the case in Cape Town. I was really interested in environmental contamination with antibiotics and other chemicals. You alluded to that briefly. Yeah. Did you measure that, and do you have a sense of how important that would be? And then, kind of related to that, you focused on one organism and one resistance mutation. Are you also looking at horizontal transmission through mobile genetic elements, etc.? Do you think that environmental bacteria, for example, or other non-pathogenic bacteria are important in the kind of One Health rural farming context or livestock human interface context?
Yeah, so I'll do my best to answer all of that because they're all really good questions, but quite different. So going back to your first question, we focused on two bugs, E. coli and Klebsiella, not because anticipating that they would have different biologies, but that this was very expensive work to do, and that's why we decided to zoom in, and we did decide to zoom in on human factors rather than looking at conjugative transfer of mobile genetic elements. Sorry, I didn't show the data. We published 2 papers in the last couple of months looking at potentially resistance driving chemicals in the Blantyre environment, and I'll share the links to those with Elsa to forward to you. We found incredibly high levels of trimethoprim and sulfur methoxazole in environmental samples and reasonable levels of metronidazole. We didn't find a lot of amoxicillin, but that is because my understanding is amoxicillin does break down very rapidly after contact with the environment. There's also lots of other chemicals in the environment which we would predict as being highly likely to drive the kind of conjugative transfer you describe. And of course, it's warm in Malawi, so there is a favorable ambient temperature to support conjugative transfer. So yes, I think the fact that those chemicals are going into the environment, and again, that there isn't that separation of humans from waste and similarly animals from waste is potentially a really critical driver. Part of what inspired this study was a study I led 10 years ago into a major typhoid outbreak in Malawi, which started in 2012-13. And one of the key risk factors in that was not drinking water. It was all of the other water that people used in their activities of daily living. So it was use of river water for purposes other than drinking. And in some of those photos, you'll have seen people washing clothes and also washing kitchen equipment in heavily contaminated river water. So I'm absolutely certain there is conjugative transfer going on. In answer to your question as to what role do secondary pathogens play, it's a really interesting question. And I think what probably wants doing at some point is for someone to write a grant, and someone may have done this and I'm simply unaware of it, actually constructing a model system that can be really tightly controlled and perturbed to look at the impact of different chemicals in the environment on those drivers. Helen, did I answer all of your questions? Because there were several ones.
Yes, thank you.
Thank you for your question.
Interesting.
Thomas, you can carry on.
Hi, thanks. That was really an amazing talk and amazing work you're doing. I have kind of 2 questions. The first one kind of relates to your work or to the slides where you show like these 3 different regions in Malawi where you see like kind of a high burden of antimicrobial resistance in some of these areas. I was just like wondering because kind of in one of these murder trial papers where they also kind of exposed children to azithromycin, they were kind of they saw like this spillover effect which no one really can explain, but one of these possible explanations is that it's like these people are all along kind of a river or wastewater kind of line. Is this similar to what you're seeing? That because I mean, it's also a little bit astonishing. A lot of people were not really exposed to antibiotics and resistance kind of threats. Is this kind of all across like along a river or a wastewater line?
Yeah, so I think that's certainly possible. People or most households interact with the environment in a way that would be unusual in the UK, and we saw that the bugs are viable in those samples. It was very easy to pull them out. So I do think people are probably regularly ingesting them. So there's an ongoing piece of work using an approach which my water sanitation and hygiene colleagues refer to as the construction of shit flow diagrams to try to look at that kind of gradient and think about whether spatial modeling can be done to look at the kind of density of infection upstream and downstream. And that work is ongoing, and it will be helped. There's still a few more whole genome sequences to analyze, and it will be helped when we can start to characterize that and again reconstruct transmission networks up and downstream. Does that answer your question, Thomas?
Yeah, no, that's perfect. That's really, really interesting. And the second question is, you showed that kind of all households had at least like one time point where at least someone had like a carriage of ESBL. Did you look kind of what are factors that people kind of lose it over time, or also factors that some people in the household, assuming that they had more or less the same exposure, didn't get it at all?
It's something I'm very interested in looking at. So there is a study that Patrick, who I mentioned a couple of times, is now leading through a Wellcome International Fellowship, and he's trying to recapitulate Joe Lewis's experiments. And it may be that there is a signal that we're seeing that the people who come into hospital with an ESBL E. coli are the same people who go from home from hospital with an ESBL Klebsiella having been acquired. But even then, that's interesting because Klebsiella naturally lives further up the intestine, and it might simply be that the ESBL Klebsiella was also already there. But actually, under this favorable selection pressure of antimicrobial usage, it just migrates further down to the point where stool culture actually has the sensitivity to pick it up. So it is complicated by the fact that when we take a stool sample, we are only capturing a tiny amount of the 20 feet of bowel, which I think limits a little bit the extent to which we can do that kind of modeling.
Okay, thanks a lot.
I see we also have a question from Alani Suleiman.
Thank you very much. I just want to be revive study specific. The antibiotics here is azithromycin, and it's like for most bloodstream infections, whether CLEB or whatever we're talking about, the antibiotics that only develop resistance is the one that produce the beta-lactamases. Azithromycin appears, I'm just suggesting, can we expect that azithromycin will be very effective against those organisms that produce the beta-lactamases? Are we confident that in the study we are carrying out, we should not expect a large number of our patients to develop any form of resistance, assuming that the BSI will be expected. We can only produce beta-lactamases, and azithromycin will still be very effective in the presence of beta-lactamases. I just want your opinion on the study that we are conducting.
Thank you. I mean, I think one of the really interesting things about clinical microbiology is so much of it is based on practice and experience, and an awful lot of the early days of antimicrobial usage were not subject to rigorous randomized controlled trials, and so that there are huge yawning gaps of infection management. There are lots of people who are working very hard to close those gaps now, but there is nothing like a trial to inform a change in practice. So working on mechanistic data, not having the benefit of trial data, my concern about azithromycin is that so much of it is concentrated in the intracellular compartment that it's not an ideal, I would guess that it's not an ideal agent for the treatment of any bloodstream infection other than typhoid, which of course is also something that sits in the intracellular compartment. So it may well be that I'm wrong, because what I'm basing my information on is the parameters of the drug rather than a clinical endpoint. But I think that would be my concern about azithromycin for the management of bloodstream infection. But that might just be because I'm very old-fashioned, and the trial will give us the information we need. In answer to your question about co-selection, there are some antibiotic resistance genes which are classically on the same cassette, which undergoes conjugative transfer as the CTXM15 cephalosporinases. So you might expect to see chloramphenicol. Well, in Malawi, actually, the ESBL gene has dropped into a chloramphenicol acetyltransferase. So we've started to see the re-emergence of chloramphenicol susceptibility in Malawi. So there's quite often a disrupted chloramphenicol acetyltransferase, resistance to trimethoprim, resistance to cotrimoxazole, resistance to gentamicin, and resistance to aminopenicillins will all cluster together. I'm not aware, but again, this may be my own ignorance, of azithromycin resistance clustering with those particular resistance determinants. So I would perhaps be less concerned about co-selection of beta-lactam resistance by use of azithromycin than I would about having an anxiety about the role of azithromycin in the management of a sepsis syndrome.
Many thanks.
Thank you.
Nick, maybe I can just add, from the side of the REVIVE trial, Alani, is, that we would not suggest azithromycin be used as treatment for bloodstream infection, and the rationale for which we're using in the trial is not as treatment, but as prophylaxis against the development of infection, which is quite a different thing from treatment. We also not making any determinations or statements that it would have very broad coverage. We feel that it has coverage against certain important bacterial pathogens that affect people with advanced HIV disease, particularly pneumococcus and salmonella. But there are many other pathogens for which it won't cover. And so it's definitely not a strategy that should be used for treatment of bloodstream infection, and it's definitely not a strategy for which we think will cover all potential bacterial pathogens. But by covering certain of the important ones, our hypothesis is that will reduce mortality. So I just don't want to give the impression that we're overstating the benefit of what azithromycin might have. Alani, does that answer the question that you had?
I'm grateful for that clarification. Thank you so much.
Thanks very much.
Okay, I don't see any other questions. Let's just check. If you would like, with Prof. Feasey's permission, we will be sharing his presentation recording on the REVIVE website. But if you would like direct access, you can also just drop your email address in the chat or email me directly, and I will share it with you after this presentation. And then, Prof. Feasey, we would like to thank you for an incredibly insightful talk. I think we could keep you online for much longer, but we won't. So thanks for your time, and thanks for this. And I'm sure that we will have you back for another follow-up talk. I thank everyone who was joining, and may you all have a very blessed week further.
Thank you for having me.
Thank you.
Thanks very much.
Thank
Nick.
you.
Thanks very much.
Thank you.
Thanks,
Bye.
everyone.